And feel free, there we go. If anything's unclear, feel free to interrupt me along the way. I'm happy to field questions as I go. So I'm going to talk mostly about some work that I started during my postdoc and have recently wrapped up. And there's kind of three parts of the talk. So the first part of the talk, I'm going to tell you about our efforts to apply a behavioral economic theory called prospect theory to rats. And then the second part of the talk, I'm going to describe the role of the orbitofrontal cortex in this task. And finally, I want to circle back and talk about at a high level ways of reconciling ideas of subjective value between economic and reinforcement theories. Okay, so as a neuroscientist interested in decision making, I'm drawn to behavioral economics because I think it provides a really useful quantitative framework for describing choice behavior. And I just want to go over some kind of key terminology and concepts. So the main workhorse of modern economic theory is the idea of utility. And so the idea is that we don't evaluate outcomes or rewards based on their absolute amount, but based on the subjective satisfaction or utility that we derive from consuming them. And so you can capture that by something called the utility function, shown here, which maps reward, here I have monetary gain on the x-axis, onto utility or subjective satisfaction that you get from that reward. And the particular shape of this function has consequences for our preferences and for our choices. And so canonically, it's been observed that people tend to have this shape, which is called concave, and that has implications for our risk attitudes. And just to give a concrete example, one particular phenomenon that this shaped utility function captures is something called diminishing marginal sensitivity. And so this is related to Weber-Fechner's law, right? The idea is that the difference between $0 and $1,000 feels a lot bigger to us than the difference between $110 and $111,000, even though the delta dollars there is the same, right? It's 1,000. And you can account for that by if you have this shaped function, because down here the curve is much steeper, and so those values are more separable on the y-axis. Whereas here, because it's shallower, you get a compressed representation. And so that is one aspect that's just a concrete example of something that this utility function can capture. So the predominant behavioral economic theory is called prospect theory. It was developed by Daniel Kahneman and Amos Tversky and won them the Nobel Prize in Economics. And it describes basically four main features of human choice behavior. So the first is the phenomenon I just described, diminishing marginal sensitivity for gains, and you can capture that by having a concave utility function in the gain domain. The second key feature that prospect theory describes is called reference dependence. So the idea is that you evaluate outcomes as gains or losses relative to some internal reference point that at least some of the time probably reflects your expectations. Additionally, prospect theory describes loss aversion. So the observation is that people are more sensitive to losses than to equivalent gains. And you can account for that by having a loss function that's steeper than your gain function. And finally, people exhibit what's called probability distortion, where they overweight the likelihood of low probability outcomes and underweight the likelihood of medium to high probability outcomes. And these observations describe a lot of empirical choice data in the human literature, for instance, like choices about insurance, purchasing decisions, labor, etc. And so for economists, the equations that describe these functions are a set of tools that allow them to describe human choice behavior. But as a neuroscientist, I think these are really interesting cognitive phenomena. And so we're interested in leveraging the powerful experimental toolkit in rats to try to understand their neural basis. But in order to characterize these phenomena, we needed to develop a task that would let us do so. So economic theories of choice have historically been studied in primates. Obviously, economics was developed to describe human choice behavior, and there's been really beautiful foundational work applying those same theories to macaques. And those tasks typically elicit what an economist would call our decisions from description. And so here you're explicitly offering the subject, let's say different lotteries and asking them to choose between them. And those are qualitatively different paradigms from what have typically been used in rodents, which elicit decisions from experience. So in this case, think of like a bandit task, where an animal has to learn about value from actively sampling and experiencing it. Those are kind of fundamentally different paradigms from decision from description tasks. And so we wanted to develop a task that would allow us to, in rats, elicit decisions from description. And I'm going to describe in a moment how we did that. But briefly, our approach was to use high throughput behavioral training. So an advantage of rats and rodents in general is that they're small and they're cheap. And so we can train many of them in parallel. And so this is a picture of the high throughput behavioral facility in Carlos Brody's lab where I did my postdoc. We've since developed our own high throughput facility. Carlos's lab, when I was there, was training about 300 rats a day, and the training is all computerized and automated. So once you write the software that transitions the animal through the different shaping procedures, you basically put a rat in, and then a couple months later, you get a trained rat out. And so this lets us do at least two things. The first is that this generates a lot of data, both per animal and across animals. And so this gives us a lot of statistical power for behavioral modeling. And additionally, because we can train so many rats in parallel, we can really explore individual variability in a way that can be challenging in other systems. Okay, so here's the task that we developed to elicit decisions from description. So the key thing is that we wanted to explicitly offer the animal, a lottery and a sure bet on each trial. And we use sensory evidence to do that. And so the rat initiates a trial by poking his nose in a central nose port. He's then presented with a series of light flashes, the number of which tell him the probability of reward at each port. So here, 3 flashes means probability of 0.3, and 10 flashes means a guaranteed reward on the right side. Simultaneously, there's auditory clicks, the rate of which convey the reward volume that the rat's being offered. So here, he's choosing between a gamble of 48 microliters with a probability of 0.3 or a guaranteed reward of 6 microliters. And then at the end of the trial, he's free to just choose the side that he prefers. And so a couple things to note. One side always offers a guaranteed reward. And in this particular example, that's smaller than the risky bet, the risky option, but that's not always the case. The volumes and probabilities are chosen independently. And so the probabilities go from 0 to 1 in 0.1 increments. And that was important so that we can resolve the shape of the probability weighting function. And there's 4 possible reward volumes that are on a log base 2 scale. So they're roughly equally discriminable to the end. Okay, and there's a couple, you might think that this is a difficult task for the rats to learn. It is. It took them, anywhere from 4 to 6 months. But that was okay because, I wasn't the one training them. And, that's the point of the rat factory. And this is just showing performance in the final training stage only as a function of training session. And so what we can do is we can calculate what we call the efficiency, which is basically the average expected value per trial. Relative to an ideal performer that always chose the better expected value option, or a subject that chose randomly. And you can see that, basically once the animals cross threshold performance, their performance remains relatively stable. Another way, once they've learned the task that you can look at their data, is by really exploring kind of their preferences as a function of different trial types. So this is data for one rat, and this is a very rich data set, right? So it's kind of difficult to visualize everything. So one way to do it is to plot a separate choice matrix for each guaranteed or safe side volume. So this matrix shows the animal's choices, where each block is the percent that the rat chose safe for trials offering a particular guaranteed reward, here 6 microliters. The y-axis is the volume of the risky reward, and the x-axis is the probability of the risky reward. And so these are the choices for 6, 12, 24, and 48 microliters on the safe side. And so just a couple things to keep in mind: when the safe side is the best option, this matrix is almost white because the animal is choosing the safe side almost all of the time. Whereas if the safe side is the worst option, 6 microliters, then it's pretty red and the animal is choosing the risky side more of the time. And so this is for one rat. And this is averaged over 36 rats that we trained on the task. Okay, so this is just to show you, kind of to unpack the data a little bit and to drive the point home that we have a really rich description of the animal's choices across a large choice set. But one thing, of course, that we worried about is that the animals didn't really understand the meaning of the cues, and they were using some perceptual heuristic, for instance. So one piece of evidence that suggests that that's not the case is shown here. So the rats were supposed to keep their nose in the center port, until the end of the Q period. But sometimes they withdrew their nose prematurely, and we call those violation trials or opt-out trials. And it turns out that they didn't do that uniformly across trial types. Here I'm showing for one rat, the probability of violating or opting out as a function of the reward offered on the risky side and the safe side. And you can see when both sides are offering the smallest rewards, the animal's opting out 30 to 40% of the time, which is quite high. But when the animal's offered the best rewards, he's basically never opting out. And so it seems like that they're basically aborting trials that they don't think are offering good enough options. And this is really robust across rats. Even if we disproportionately penalized violating on these low volume trials, we weren't able to train them out of it, which was kind of annoying, but also encouraging because it really demonstrates that they understand the meaning of the cues and weren't using some alternative heuristic strategy. Okay, so I apologize for the equation. If you're not into it, I just added this at the last minute and I'm not sure if it's effective or not. But if anyone, if there's any aficionados in the audience, we basically used a fairly simple behavioral model where we computed the we assumed that the value of each offer was the product of the subjective utility and the subjective probability of the gamble on that side, where this weird function parameterizes the probability weighting function and the utility function is the reward to some power alpha. And then the difference in the value on the right and the left side is the argument to the logistic function, and that's how we get a choice probability. And so we have a lot of data, and so we're able to, the model is identifiable and we can recapitulate the data with the model fits. So here on the top row, I'm showing those choice matrices for one rat, where now it's 6 microliters, 12, 24, and 48 on the safe side. And this is just to show you that the data looks like the model, right? So the bottom row is basically the model prediction of the rat's choices on held out test data. And you can see that, qualitatively the model's capturing the rat's behavior. And so what's nice is that now the model is able to recapitulate the rat's data, and now we can ask, okay, what does the utility function and the probability weighting function look like from the model? And so across 36 rats, this is what we see. So the rats tended to have these concave utility functions like humans, and so that produce something like diminishing marginal sensitivity, which I mentioned in the beginning. And they also tend to distort the probabilities, although most of them don't look like the kind of canonical inverted S-shape function in humans. They tend to, in general, overweight the likelihood of most outcomes. So that's interesting. I don't really have time to go into all of the ways that we tried to kind of independently validate this, right? So whenever you have some results from a model, you don't want to rely too much on the fitting. You want to also do kind of model agnostic corroborations. I'll tell you about one briefly, which is that there's a prediction if the animals have this particular shape, which I mentioned, is that they should exhibit diminishing marginal sensitivity. And so we can test that directly in the following way. So if you had a linear utility function, then the difference between 24 and 0 should be equally discriminable to 48 versus 24, right? Because those are going to have the same distance on the y-axis. But if you're concave, then 24 should be more different from 0 than 48 is from 24. And so you can just look at those trials that offer those two options, guaranteed rewards of 24 versus 0 and 48 versus 24. And if you're concave, you should be choosing the better option more on the green trials than the purple trials because they're more discriminable. And so this is what we find for one rat as an example. And moreover, the relationship between the power of the, how concave your utility function is, where smaller numbers mean more concave, is directly correlated with how much better you are on the green trials compared to the purple trials. And if you have basically a linear utility function where your alpha here is 1, then you're doing equally well in these trial types. And so this was one way that kind of gave us confidence in interpreting the shapes of the utility functions from the model. Okay, so briefly, I've shown you that rats exhibit these two features of prospect theory, and then I just want to kind of briefly touch on our evidence that they also exhibit reference dependence and loss aversion. So reference dependence is the idea is that you're evaluating outcomes relative to some internal reference point. And so this became apparent when we looked at their trial history effects. So here the rats exhibit these sequential biases, which can be quite strong in the spatial domain. So here I'm plotting the change in the probability of repeating a left-right choice relative to the average probability of repeating, conditioned on whether the previous reward was either an unrewarded trial or one of these volumes. And so you could see when the animal gets 48, he's more likely to revisit that port. So that's a win stay bias. And if he doesn't get reward, he's more likely to switch. So that's a lose switch. What's interesting is that this curve crosses 0 at 12, right? So if the animal's getting 6 or 12, they're more likely to switch to the other side, which suggests that they're treating those values as being in the loss domain. And you might worry that, well, maybe the solenoids are unreliable at those volumes. And so for 3 rats, for their lifetime, they received rewards that were all doubled. So they were rewarded from 12 to 96. And now they're switching after 12 or 24 microliters. And so this seems like they're basically subjectively treating these different rewards as losses relative to the average. And moreover, where that threshold is reward history dependent. So here for one rat, we're plotting the same win stay lose switch curve, but conditioned on whether 2 trials back the animal was rewarded or not. And you can see that this shifts. And so we think that the animal is using kind of its local reward history to compute something like an economic reference point. And there's a prediction here, which is because the utility functions are concave, their performance should be better when the reference point is high. And just to give an intuition, that's because when the reference point's high, you're basically pulling the rewards down to a part of the curve that's more discriminable, right? And indeed, if we just plot if we compare the slope of the psychometric curves when the reference point is high compared to when it's low, we do see kind of steeper performance when the reference point is high. And this is for one rat, but we see this across the population of 36 rats. Okay, and at the aggregate level, this is the median value function for all of the rats. And we did see a steeper slope for the loss function compared to the gain function, consistent with loss aversion. So in this current biology paper, we basically showed that the rats exhibit all of the key features of prospect theory. And that kind of is really exciting because it means that we can leverage the toolkit in rats to start to interrogate the neural basis of these phenomena. And my lab is really focusing on reference dependence at the moment. Okay, so now I want to switch gears and talk about the neuroscience. And we specifically focused on the orbitofrontal cortex, both because it's been implicated in economic value or value-based decision-making, and also because of the trial-by-trial effects that I'm going to talk about a little bit more. It's also been implicated in updating values from experience. So I briefly mentioned the Wednesday lose switch bias. The rats exhibited multiple types of deleterious trial-by-trial learning. And I say deleterious because in this task, the outcome on each trial was independent. And so using things that happened on previous trials to update your strategy is actually bad. But the rats nonetheless do it. Humans do the same thing. And so an additional bias that the rats exhibited is what we ended up calling the risky win stay bias. So they're more likely to gamble following risky wins. And so that's shown here. This is the change in the probability of choosing the safe option relative to the average probability of choosing safe, conditioned on whether the previous reward was a safe option or a risky choice. And you can see all the rats basically are less likely to choose a safe option if they gambled and won. Moreover, there's a graded dependence on the probability of the risky reward. So if on the last trial I chose a gamble and it was really unlikely to win, but it did, then I'm even more likely to choose the risky side on the next trial. And so this also kind of confirms that they really understand the meaning of the flashes conveying the reward probabilities. So this effect is much smaller than the spatial bias, right? The other one was on the order of about 10%. Here we're talking about a change of 2 to 5%, but nonetheless, they exhibit this bias as well. And this has also been observed broadly in humans and also macaques. Okay, so there's two types of sequential effects that I've described. There's spatial and a risky Wednesday bias, right? So we can dissociate them because the safe and risky ports switch randomly on a trial by trial basis. So let's say on trial I, the rat chooses the gamble, which happens to be at the left port, and gets rewarded. On the next trial, he can either revisit that spatial location, and so that would be, or have an increased likelihood of revisiting that spatial location, and so that would be a Wednesday bias, or he could have an increased likelihood of choosing the lottery or the gamble, and that would be a risky Wednesday bias. And again, we see these, we're able to dissociate them, and we see both of them contributing to behavior. Okay, so in terms of the place that we decided to kind of stick our electrodes and investigate, we looked at the rat orbitofrontal cortex, which is part of the ventral PFC. Specifically, we're in kind of the lateral OFC. So primates, really interesting, have a really elaborated granular prefrontal cortex that's absent in the rat. We think that we're somewhere around the caudal third of the monkey OFC, which is also a granular, probably area 13, and that shares kind of long range inputs and outputs with area 13, and also similar results from lesion studies on behavior, such as devaluation paradigm. But if people want to kind of talk about species homology, I'd love to chat about it. I think it's really complicated and interesting. But anyway, we think that we're in rat LO and some anterior insula, which is probably something like this. Okay, so OFC is part of the prefrontal cortex. And so I want to talk about our efforts to kind of unravel some of these really complex representations. This is work done by a talented postdoc in my lab, David Hocker, who's jointly advised by Christina Savin. Christina is in the Center for Neuroscience and also the Center for Data Science at NYU. And this was recently published this past month. Okay, so there's kind of two different, very strong hypotheses about what encoding should look like in the prefrontal cortex. And you can think about this at the level of individual neuron responses or populations. So let's start with individual neuron responses, right? If you have a task which is complex and there are multiple variables that might be encoded, one possibility is that you have what's called mixed selectivity, where individual neurons can be tuned to different, here, these are stimuli, to different aspects or behavioral variables. And there's evidence, this has been widely reported in recordings from frontal cortex and has some specific advantages for being read out by downstream circuits. The alternative hypothesis is that you have neurons that basically have pure selectivity and are only responsive to one aspect or behavioral variable in a task. At the level of the population, you might have randomly distributed encoding. So you have neurons that will basically encode this entire space of possible responses across different task variables, or you might have distinct subpopulations or clusters of neurons that encode different stimuli or task variables. And so if you combine these together, these are basically kind of competing hypotheses about representations. Here I have an OFC, but it's really about prefrontal cortex. And there's data supporting both of these, right? And so one idea is Randomix Selectivity, which I mentioned, where you have individual neurons that span the full possible response space of different task variables and are encode information about different stimuli, or you have different subpopulations of neurons that encode particular decision variables. And work from Adam Kepish's group, for instance, found evidence for this case, and that particularly one of these clusters projected to a common downstream target. So we were wondering if we could parse or test these hypotheses in our own data. So the first thing is, if we want to test for whether or not there's clustered responses, we decided to do k-means clustering on just the time average PSTHs averaged over all trials. So here, these are the, each row here is a neuron, and this is the average firing rate for that neuron aligned to trial initiation. And it looks like, maybe there's some cells that are responsive at trial start, and then a bunch out here that are responsive around the time of the choice. But it's not obvious that there's much structure. But if instead we replot these PSTHs grouping together neurons that have been clustered together by the clustering algorithm, we could see that there is there are stereotyped responses aligned to different task events. And so I'm happy to get into kind of the statistical ways that we made choices about, for instance, how many clusters, but you can also refer to the paper if you want to really get granular. But we found evidence for 5 clusters. This red cluster is the only one that really was active during the Q period, and most of the others were active either at trial start or after the choice was made. And so this was really striking and interesting, right? That what looks like a mess and what we might have missed is actually hiding kind of real structure and low dimensional structure across these 5 groups. And this is showing just the average z-scored firing rate as a function of cluster. And so, as I mentioned, cluster 2 is the only one that's really active during the Q period, and cluster 3 has this really prominent response at reward. So a different way to cluster is, instead of just using the average PSTH, which just tells you when in the trial is the neuron active, you might want to do something smarter and ask, okay, well, what does the neuron encode? And if we clustered on that, maybe we can also learn something interesting about the organization of responses. And so for this approach, we developed what's called a feature space. And so it's basically a 19-element vector, where each element of that vector is the firing rate of the neuron on a particular trial type. And so here, let's say the first element is the firing rate on trials at the time of reward if the animal wins versus if the animal loses. Then you also can take into account reward history, etc. And so this is a way of basically having a compact representation of how the neuron responds to these different task variables. And if you cluster on those conditional feature spaces, it's interesting, we saw very similar results. So again, we identified 5 clusters that in some ways look very qualitatively similar to the clusters that we obtained from just the average PSTHs across all trials. Interestingly, the neurons in the red and green clusters tended to be were conserved across both clustering methods, suggesting that those neurons both exhibit similar PSTHs or temporal response profiles, and also encoding as defined by the feature space. Okay, so then the question is, do neurons in a particular cluster have specific types of responses? And so to get at that, we built an encoding model, or a GLM, which basically tries to capture how the neuron responds to different events in the task. And it does that by having these response kernels that are convolved with either stimulus events, so here are clicks and flashes, or behavioral events like choice and reward outcome. And the firing rate of the neuron is the exponentiated sum of those response kernels on a single trial. And then we can use a Poisson generation process to get spikes. And so the GLM does a good job of capturing the average PSTHs of the neurons. These are 5 example neurons shown here. And so then we can ask, well, what do the kernels look like? And specifically, what we're going to do is we're going to compute a metric called the coefficient of partial determination, which quantifies how much a particular response kernel improves the goodness of fit of the model. And so let's say I cared about how much this neuron encoded reward. What I would do is I would have my full model that includes a reward kernel, and then I would take that reward kernel out and look at how the difference in the variance explained, or how much worse does the model do without the reward kernel. And that's a way of using the GLM to quantify the encoding of these different task variables. And so the question is, do certain clusters encode different certain task variables? And somewhat disappointingly, the answer was no. So here I'm showing, this is the CPD, or the percent variance explained, that's accounted for by the reward history kernel. And so this is basically a kernel that says the previous trial was rewarded. And we knew from just the raw data that this tended to be encoded around the time of trial start. And so we see that in the CPD, but this is the average CPD as a function of time for the different clusters, and they all look pretty similar. Similarly, they all have pretty strong encoding of left-right choice around the time of choice. They all have pretty strong encoding of reward. And they all have encode both flashes and clicks during the Q period, although it's, one thing to note is that the magnitude of CPD is an order of magnitude lower than for the other covariates, right? So this is basically, these are on a different y-axis. And so there's actually very weak encoding of the stimuli that convey reward attributes, which was pretty surprising and, not what we were really expecting. So we didn't see very strong encoding of the reward attributes that the animal would be using to drive the choice. And instead, the strongest encoding that we really saw occurred after the choice had already been made, which is consistent with what other people have reported in rodent OFC. And so based on the neural recordings, we didn't necessarily think that there would be a strong effect of OFC on driving the choice on that trial because we didn't see strong dynamics that reflected the deliberation process, for instance. Instead, our hypothesis was that these dynamics that occurred after the choice had been made might be important for some of the sequential trial-by-trial learning effects that we observed. And so our hypothesis was that these representations could either subserve the risky win-stay bias or the spatial win-stay loose switch effects that we observed. But just looking at the encoding, it wasn't obvious which one, right? So to test that, we used optogenetics. So we bilaterally expressed either halorhodopsin downstream of a promoter for excitatory cells or chanorodopsin downstream of a promoter for inhibitory cells. And then we inactivated for 4 seconds, triggered off when the animal left the center port, and then looked at behavior on the subsequent trials. And this is just to validate, at the time that we did this, I'm not sure other people had used these PV-Cree rats for optogenetics, so we validated that we specifically get expression in the PV cells, and also that we get inactivation in vivo. Okay. So when we did this, we saw that photoinhibition of OFC eliminated the risky win-stay bias across all groups. So this is the halo excitatory cells. And we see a reduction. We see reduction in the PV-Cree excitatory cells, and no effect in the sham animals. I do want to point out that this is a small effect size, right? But the magnitude is limited by the magnitude of the risky win-stay bias to begin with, which is small. And it's also a robust effect that we saw across 13 rats and 2 manipulations. So even though it's a small effect, we feel pretty confident about it. So anyway, we eliminate the risky win-stay bias on the next trial. And when we first looked at the spatial win-stay bias, it looked like we might also have an effect, right? So if we compare the bias following reward, there's less of a probability of repeating on the laser trials, less of a probability of switching on the laser trials. But those effects were also present in the sham rats, which was kind of intriguing. And so to understand this further, the issue is basically that because we were inactivating during, after the choice had been made, the opto trials imposed a minimum intertrial interval of 4 seconds. And because the trials are self-paced, this is the distribution of intertrial intervals for a single rat. And you can see most of them are actually less than 4 seconds. And so this effect is because the bias itself is decaying with time. So if we take that into account and we only make these bar plots for long ITI trials, then there's no difference. And so the photoinhibition didn't have an effect on the spatial win-stay bias. But there's also kind of another thing which is intriguing, which is that the spatial bias seems to decay with time, whereas the risky bias does not, right? Because there's no effect on the sham rats. And so that further argues that these different trial-by-trial learning effects are dissociable and mediated by distinct neural mechanisms. Okay, so we were intrigued about which kind of subpopulation of OFC neurons might mediate this particular effect. And around the time that we were doing this work, this paper from Adam Kepish's group by Hirakawa et al. Came out. And it was pretty exciting because they basically optogenetically tagged OFC neurons that projected to the ventral striatum and found that they exhibited a particular kind of pattern of conditional responses. So after the choice, their OFC to striatum neurons had higher responses following unrewarded compared to rewarded trials. And then leading up to the subsequent trial, there was kind of a graded representation of what they called negative value, because there were higher responses following unrewarded trials and lower responses following high rewards. And so we basically plotted the average PSTH of our different clusters, breaking up the trials in a similar way. And interestingly, we found that cluster 3 was the only one that kind of qualitatively looked like the OFC to striatum cluster. So it was a different task. And so what's going on here is a little bit different, but in general, we see negative value coding after the choice. And importantly, this was the only cluster that had persistent negative value coding until the next trial. And so this was super exciting because, you know, it's really hard to replicate things. And, you know, this is a different lab using a totally different task. And yet we were able to kind of identify the same cluster, you know, a cluster that had qualitatively similar responses to their cluster that projected to the striatum. So that, you know, suggests that cluster 3 might include neurons that project to the striatum as well. And so if we looked a little bit deeper at cluster 3, here, this is the coefficient of partial determination. I'm sorry, these should be in percent, so multiply everything here by 100, of reward history. So basically, when the animal starts the next trial, there's still a memory of whether the previous trial was rewarded that's being expressed across the population. But cluster 3 is the only one that kind of has that reward history representation reemerge at the time of choice. And so one possibility is that provides a way of kind of mediating ongoing, influencing ongoing neural dynamics that are driving the choice. So these reward history representations might have a direct effect on how reward history affects the current choice. Another possibility is that, corticostriatal projections are thought to reflect the value of different states in a reinforcement learning framework. And so having neurons that project to the striatum reflect reward history at the same time as it reflects other contextual inputs like reward and choice might allow for kind of richer state representations in the striatum, if that makes sense. Okay, so to go back to our hypotheses about encoding, we had, these very strong hypotheses of either Randomix selectivity or functionally distinct subpopulations. And we think we found something that's somewhere in the middle, where we do see clustered responses, but within a particular cluster, we tend to see mixed selectivity. And the thing that kind of separates these clusters is the temporal response. And so it might be that you have neurons that are active at different parts of the trial because they're contributing to behavior in a particular way or communicating with the downstream circuit in a way that requires some temporal precision. And so our hypothesis is that, maybe these neurons that have different temporal response profiles are talking to different downstream targets. And we're now doing optogenetic recordings of different projection-specific populations in OFC to kind of test this more concretely. Okay, so the part 2 conclusion is that neurons in OFC belong to one of a small number of distinct subpopulations or clusters. Perturbations of OFC eliminate abstract task-specific biases, but not spatial ones. And putative corticostriatal projection neurons seem to uniquely encode reward history at the time of choice, which could either reflect that they are actively influencing dynamics driving choice or providing richer contextual inputs tagged for plasticity in the striatum. Okay. And so the very last part of the talk is going to be pretty brief, but I just want to kind of synthesize the first 2 parts, which are, a little bit at odds in terms of how to think about subjective value. So in the first part of the talk, we applied prospect theory to rats behavior. And in general, in economics, the idea is that subjective value is determined by the shapes of these subjective functions, right? So you have a utility function, really this should be a value function, and a probability weighting function. And that these can vary across individuals, but are fixed for an individual. And so economics, this standard kind of economic framework does not account for learning, which, as I showed you in the entire second half of the talk, is a main feature of the animal's behavior. And that's actually at odds with animal learning theory and reinforcement learning frameworks that account for value. And so, for instance, let's imagine that we had Pavlov's dog that learns to associate a conditioned stimulus with a reward. Learning theory described initially how the animal might build stronger associative, or might refine its associations between the stimulus and the reward by updating the associative strength, here A. And those models were eventually extended to account for how animals might not just learn associations, but also learn values directly, where they're updating their previous value based on the reward outcome, where this value corresponds to a reward prediction error. And so there's kind of one subtle issue here, which is that this equation and similar equations will learn the expected value of a reward. So let's say that the pairing between the bell and the steak wasn't deterministic, but was probabilistic, and that on 50% of the trials, the bell resulted in the steak, but on the other 50 it didn't. That would mean that on half of the trials, R would be 1, and on the other half, R would be 0. And so with enough data, the V of t would converge to 0.5 times R, which is the expected value. And that necessarily implies linear utility and probability weighting functions. And so just to make that point directly, we simulated the choices of a Q learning agent on our gambling tests that I described. And then we fit the prospect theory model to the agent's data. And sure enough, this is really just kind of a demonstration, right? But we recovered linear utility and probability weighting functions, which are not consistent with the animal's behavior. And so we propose a very slight tweak to the framework, which is that if you had a reinforcement learning agent whose subjective value or whose reward prediction error was in units of subjective value according to prospect theory, then you can basically account for all of it. You can account for the trial by trial learning effects, and you can also account for the nonlinear utility and probability weighting that we observed in the data. And we played around with a number of different reinforcement learning agents, but an agent where the RPE, or the reward prediction error, is in units of subjective value compared to just the objective reward outperforms for all the rats. And this is not, so I would say two things. First, we think that this is biologically plausible and also not our own idea. And so there's a wealth of evidence that suggests that dopamine instantiates a reward prediction error and a temporal difference learning algorithm in the brain of the kind formalized in these animal learning models, or learning theory models. And there's evidence that that RPE is in units of subjective value. So this is kind of a classic paper from Wolfram Schultz's group, where they have a stimulus that's going to come with a different delay. And this is the firing rate of a dopamine neuron after a cue kind of instructs the animal about the delay that the reward's going to come. And you can see that there's a higher peak for short delays, and then progressively smaller peak for longer delays. And the reward prediction error here reflects the hyperbolic temporal discounting of the monkey. And there's more recent work also from William Stouffer and Wolfram Schultz that further suggests that the reward prediction error might be in units of subjective utility. And so we agree with that hypothesis, and we think that this is a parsimonious way of reconciling these formal economic models with reinforcement learning. Okay, so in summary, I feel like I got through everything, which is very surprising. I've shown you that rats behave according to prospect theory, the predominant behavioral economic model. Orbitofrontal cortex promotes trial by trial learning, even when it's maladaptive. It also specifically promotes learning in abstract task-specific coordinates, but not spatial ones. And at a high level, we think that a prediction error in units of prospect theory value could integrate reinforcement learning and economic theories of choice. And so most of this work was done in the Brody lab at Princeton, and then the most recent eLife paper was done with David Hawker and Christina Savin at NYU. And there's David. This is when we went out for tacos and margaritas. And so this is my wonderful lab, and I'd like to thank them and thank you for your attention.
Thank you, Christine. That was an amazing talk. Yeah. So if there's any questions, you can either put it in the chat or you can like unmute and yeah, go ahead if anyone has any questions. I guess I was interested in the clustering methods. So like initially it was like I like all the clusters were like it'd be cool to see if they exist after all, because I think the paper it reveals more about whether they're present, not just like, you know, like more about profiling them, but like not looking at whether they're present versus absent. So I was just wondering if you had considered that.
Yeah, that's a great question. So we actually did do a statistical test to ask whether or not there was clustering present in the data versus not. And so for that, we used something called the Pair Statistic, where we basically, I don't know if I included this in my, hold on, let me stop sharing for a moment. I don't know if I included this figure in the extra slides. That's a bummer. But basically you have your like feature space, right? And you can plot the neural response as a vector in that feature space. And then you can compare the angles of nearby neurons in the feature space. And we care about the angle as opposed to the length of the vector, because we don't care about the absolute amount of firing. We care about kind of recruiting, right? And so the idea is that if you're clustered, then your angles should be pretty close together. But if you're totally unclustered, then your angles will be, you know, broader. And we compute the distribution of angles for all of the neurons relative to some null distribution of Gaussian noise. And, you know, you could just do a comparison of the medians of those distributions. And so that statistical test was the distribution of angles of our neurons was different from that of a null distribution, indicative of true clustering existing in the data. Yeah, I feel like I butchered that explanation a little bit, but it's in the paper if you want to read about it.
Thank you. I guess we have 2 questions in the chat, but I guess Alessandro, you can unmute and ask this.
Okay, thanks. So a really nice talk, very cool. I have a few questions, but I will start by one about the behavior of the rats. So do you find any, in general, any aspect in which they do differ from human economical behavior? So for example, you may know that in many monkey tasks, people find that they tend to be risk-seeking, which is different from what humans usually do. And I heard that one explanation could be that these kind of tasks have a lot of very small gambles, which may incentivize being risk-seeking. Mm-hmm. So you have any intuition about that in rats?
So the way that so I would say there's a couple ways that they're different from humans. The first is the shape of the probability weighting function, which I can speculate about. You know, so the classic shape is the inverted S shape, which Kahneman and Tversky initially hypothesized might reflect diminishing marginal sensitivity relative to the bounds of 0 and 1. But our rats kind of look like they're just anchoring to 0 and not anchoring to certainty. And I think that that might reflect a limitation of our task design, where the guaranteed reward wasn't like, we didn't have a qualitatively different cue that indicated you will definitely get reward. And so I think that the rats might just have not appreciated that the guaranteed reward was like really guaranteed, if that makes sense. And so if I had to guess, that might have reflected why we didn't get the inverted S shape. But you know, I really struggled for like more than a year to find a task that they could learn that kind of checked all the boxes. And so I tried one where there was a cue that was qualitatively different for the guaranteed reward, and I couldn't get them to learn it. Anyway, so that was one difference, is the shape of the probability weighting function. The other, which you mentioned, is the risk attitudes. And so, you know, in expected utility theory, the shape of the utility function is sufficient to account for risk attitudes, right? And it's true that in monkeys, they tend to have convex utility functions. In prospect theory, it's a little more complicated because the elevation of the probability weighting function is another way to account for risk attitudes, right? So if you're just like overweighting the likelihood of all gambles, then you're also risk seeking. And so to get around that, we computed what's called the certainty equivalent, which is basically a model agnostic measure of risk attitude. And so the idea is for a particular gamble, what is the guaranteed reward that you think is equally valuable to that gamble? And so if it is less than the expected value of the gamble, then you are risk averse because you're taking a smaller amount to avoid the gamble. And if it's greater than, then you're risk seeking. And so you can, basically we can measure the certainty equivalence for all gambles of 48 microliters, and you get a curve. And then we just computed the area under that curve to get a scalar metric of risk attitude, right? So if this is less than 0.5, then I'm systematically underestimating the gambles and I'm risk averse. Where if it's greater than, then I'm risk seeking. And so just a couple things. We both measured the certainty equivalence empirically, and then we also derived an analytic expression that predicted what those certainty equivalents should be given these parametric fits. And we see a nice correspondence. These are cross-validated, right? Where the blue data is separate from the data that was used to fit the parameters from the red curve. And across rats, we see quite a distribution actually of risk averse and risk seeking. And so I don't have a good explanation, about why it looks different from the monkey data. It might just be a sampling issue. It might be the magnitude of the reward sizes. Our rats are really also overtrained. Another idea, so, from Paul Glimcher's lab, there's this paper from Yamada that, to my knowledge, is one of the only ones that did show monkeys with concave utility functions. And they characterized them over the course of training and found that earlier in training, when they weren't, when they didn't really seem to understand the meaning of the cues completely, they seemed risk seeking. And then once they understood things better, they were risk averse. And so that could also be, that's an extremely long-winded answer to your question. But thank you for the question.
Yeah, thanks. I have a few others, but I think maybe the other people can in the chat should ask first.
Okay.
So I see a question from Kevin. Do you want to unmute and ask? Okay, I guess I'll just ask it. So Kevin asked, so is OFC involved at time of choice, but not at time of feedback learning? So
the inactivation that we did was spanned the time of choice and also feedback. So it was 4 seconds after the animal left the port and then poked into the, that included both of those. So we haven't done kind of a more fine scale, assessment within that epoch. My suspicion is that, so I mean, there's kind of I guess a controversy in the field about the extent to which OFC is important for like economic choice per se, or incorporating new information into economic choice, and specifically between Jeff Schoenbaum and Camilla Padoa-Schioppa, there's kind of competing evidence in either direction. So I didn't find in this particular task strong evidence that OFC was important for driving choice on the current trial. The consistent effect that I saw across the 13 rats was that if I perturbed during the feedback period, I prevented the learning effect on the subsequent trial. And so that's currently kind of the hypothesis that I favor, but I can't speak to evidence. As far as I can tell, there's evidence supporting both.
So we have another question from Shanil. Do you want to unmute and ask?
Hi, yeah, sorry, just give me 1 second. Hi. So I just really quickly went through the paper that you were talking about from eLife to look at the clustering analysis that you'd done, because I was really interested in learning a bit more about that. So I saw that you did k-means clustering, and I was just curious if you'd considered other clustering analysis like dbScan, and I know there's lots of other kernel-based methods as well.
We did play around with a couple, a limitation of k-means, but I think the trickiest part of any clustering method is how many clusters, right? And so k-means, you have to choose how many to tell it to cluster, right? And so the actual clustering method seemed to be pretty robust, but the important choice that we had to make was what statistic to use to determine the number of clusters. And so we tried a couple different ones, and our paper kind of discusses some of the differences, the technical differences between them in a little more detail if you're interested. There's like the silhouette score, which, and the gap statistic, which is what we ended up using. And then there's like this adjusted rand index, which is another thing that people use. And so we basically started with the way that we arbitrated between which metric to use in our data set was we had synthetic data that came from known numbers of clusters, but that had similar statistics of our data. And it turns out that in the data regime that we were in, the gap statistic more reliably was able to recover the true number of clusters than some of those other metrics like the silhouette score or the adjusted rand index. And the gap statistic also includes an explicit hypothesis test in its implementation. And so for that reason, that statistic, which told us 5 clusters, was the thing that we went with. And k-means was just kind of, you know, it's more straightforward. And since the result of the actual clustering was pretty robust to clustering method. Mm-hmm.
Thank you.
So we have another question from Matthias. Matthias, do you want to?
Yeah, sure. Yeah, thank you for the great talk. Very interesting. I was wondering, so the way you presented the utility function is what was in your talk very descriptive. And it was more like, this is basically how their subjective value is. And I was wondering whether whether one could explain it more from a normative point of view, like why are there principles that lead to these shapes of utility functions? And I was wondering whether whether this could be, for instance, explained by efficient coding, that the rats attribute more like basic coding resources to values that are more like frequent or more relevant to them. Right? And I was wondering whether you have thought in this direction and whether you have looked at your neural data to maybe to look into these like neural coding principles.
Yeah, that's a great question. That is something that we have not looked into exhaustively, but others have. So like Paul Glimsher, for instance, has been looking at kind of efficient coding of value for some time now, and he's developed a normative model inspired by divisive normalization and sensory systems that accounts for all of the prospect theory phenomena based on efficient coding principles, but with fewer parameters, right? So I think there's just like 2 parameters in his model. One is like a disposition parameter, and the other one, I feel like I'm forgetting. But basically, there's like, the thing that the model kind of struggles with is the shape of the probability weighting function. And so I don't think he's nailed that down yet, but in principle, he can account for a concave utility function, loss aversion, and reference dependence with like a divisive normalization model of value. And it makes sense, right? Like if you're gonna buy a house versus you're gonna buy lunch, those are very different regimes of value to be working in. And so you would want your system to be able to efficiently adapt to match the statistics of those environments. And so I think you probably have efficient coding applying to the domain of value as well.
Yeah, great, thanks. Yeah, I was wondering whether you, with the rat experiments where you can really like basically train them from like very early on, you can basically, I mean, you could really, I guess, manipulate like which values they should be sensitive to and which ones they should not. And
yeah, we have a task right now actually that is meant to kind of move the reference point around. I was telling Antara about this before the talk started, and it's inspired by one of Paul's tasks actually in humans. And so basically it's a temporal wagering task where we offer different reward volumes, and the rat reveals how much he wants them based on how long he's willing to wait for them. But then we adapt the rat to like blocks of trials where he's only offered small rewards versus blocks where he's offered large rewards. And we see that they adjust their wait time accordingly. So we get these strong context effects. And I think that that would be a great paradigm to kind of investigate this. And yeah, we're doing recordings in OFC. We have a lot of behavioral data, but I don't have anyone kind of working on that particular angle, but it's a good point and it's something we should probably do.
Great, thank you.
I guess we don't have any more questions in the chat, but Alessandra, if you have another question.
Sure, I mean, it doesn't matter, but well, for example, I was wondering at the beginning of the talk, you mentioned that your manipulation has aimed to do some sort of choice by description rather than by experience. But how do you know you can achieve that?
So yeah, I guess, well, I was basically trying to copy the monkey tasks, right? Like, which is what, and they present these, and specifically the whole task was inspired by Bill Stauver's study where they fit prospect theory and showed probability of distortion in monkeys. And they had like, visual cues that were conveying distinct reward attributes about volume and probability to the monkey, right? And so, the goal was to kind of mimic that, but in a rat. But there's kind of, maybe I'm reinterpreting your question, but there's like a first stage, which I'm ignoring, which is that the animal has to learn the mapping between the cues to the reward attributes, and that's done by experience, right? That's like definitely decisions from experience to some extent. And then in the second stage, they're using that mapping to guide their choices. And so, yes, I totally ignored and abstracted away the first stage, and you could say that's a critique of my approach. I suspect, but yeah, I mean, I guess to your broader question about how do I know, since each trial was independent, if the animal weren't using the cues, then their psychometric curves would be flat and their choice matrices would just be at 50%. You know what I mean? I just don't understand how like a fully naive animal that is learning from experience that doesn't understand the mapping, I don't think that they would show any performance above chance because.
Sure, no, I agree with that.
Okay.
Yeah. I was just thinking that maybe it's still different from what a human can do by just reading a number. It's different from, reading a number
Totally.
that you didn't learn by experience itself. You just know the meaning.
No, I see your point. Yeah. So I think that's a really interesting question. And, in the numerical cognition literature, there are these kind of two cognitive systems. There's like what's called the approximate number system, which doesn't require kind of symbols or linguistic characters, right? It's like your intuitive sense of probability and stuff, and it exists in animals and in pre-verbal children. And then there's the exact number system, which requires language, right? There is evidence that people and animals can use their approximate number system for probabilistic reasoning. And for instance, there's anthropological work for different indigenous groups that don't have lexical representations of numbers that are still capable of combining probabilities and estimating probabilities. And so I think that in the cognitive psychological literature, there's evidence that the approximate number system can perform probabilistic reasoning. But your point is well taken. Is it qualitatively different from, the reasoning that we do with symbols in the exact number system? Possibly, probably. But I don't think that's a problem that's specific to rats, right? I think that's any animal model, but it's a good point.
I guess we don't have any more questions, but yeah, thank you so much, Christine, for an excellent talk. And thank you everyone for joining. The questions are really great. Yeah, we're really excited to see what research things your group does in the coming year. So yeah.
Thank you so much. Thank you everyone for having me, and thank you, Antara, for inviting me. It was really fun. And nice.
Yeah, nice to meet you. Bye.
Okay, thanks. Bye.